I think we will get started. Thank you, everyone. Yeah, hello, welcome to our second webinar of the series, New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. In today's webinar on quality control for biological mass spectrometry. My name is Cindy Cook. I'm the project lead for the Bioprotocol webinar series. For those of you who are not familiar with Bioprotocol, Bioprotocol is an open access online journal whose mission is to make science and research more transparent and reproducible. Now, it is my great pleasure to introduce our moderator of today's session, Dr. David Fenjo. David received his PhD in physics from Uppsala University in Sweden. He then switched to computational biology, did a postdoc at Rockefeller University, and co-founded a bioinformatics company and worked at GE Healthcare. In 2010, he joined the New York University School of Medicine, where he's currently the professor for biochemistry and molecular pharmacology. He's the director for the PhD program in Systems and Computational Biomedicine and the master's program in Biomedical Informatics. He has over 30 years of experience with all aspects of biomedical data analysis in both academia and industry, and his work has resulted in over 200 scientific publications. His research focuses on applying data science methods and to analyze quantitative data and to model biological systems. His efforts to integrate data from multiple technologies, including mass spectrometry, sequencing, and microscopy, have provided a wide area of powerful tools to discover and verify biomarkers in therapeutic targets in cancer. David, thank you very much for being here, and I'm going to hand it over to you now.
Yes, thank you very much. And it's a great pleasure to introduce David Tabb today. So David is one of the pioneers in bioinformatics for mass spectrometry. And he's been in the field for a very long time, longer than I can remember almost. He started out when there were very few software tools available. So when it was really a necessity to develop your own tools. So, and he then developed numerous tools and always with a focus on forwarding his own research, but also disseminating them very early on to as large a community as possible. And this is something David has continued with, and he has developed search engines, software for both identifying and for quantifying peptides and proteins. And he's also had always a big focus on education. He's developed quite a bit of education material that's available for anyone to access. And what he's going to talk about today is quality control. And that sort of has been a part of his work for a long time. And the main reason for why quality control is important, you all know, is that if the data is not good, you won't get any good results out of it. So it's pretty simple, and so that's why it's very important. So, and he's done this work in a lot of different places, at Vanderbilt for quite a few years, at Stellenbosch, in Cape Town, and also in Paris. So he's worked in many different places. And he's also been involved in many international organizations like the HUPO PSI, which is the HUPO work group for developing standards. And definitely, I mean, quality control and standards are very closely related. So without further ado, David, please, we're very excited to hear your presentation on quality control for biological mass spectrometry.
Thank you so much. I really appreciate that introduction. David, you were one of the very first heroes I had coming to my first ASMS and seeing you as a graduate student present your research, I was like, I hope someday somebody thinks my research is worth a podium talk at ASMS. So it's a joy to be introduced by you. I'm going to go ahead to go into the slides, but I'd like to take just a quick moment on a little bit of biography. I realize that where I've lived and what I've been up to has been a little hard to follow, so I just thought I would make this little map. I spent the first almost 40 years of my life in the United States of America, having grown up in the Midwest, and I got my degree on the West Coast and then worked in Tennessee for more than a decade. But then I moved to South Africa, where I met my wife, and enjoyed 5 years in the tuberculosis community there. And at the moment I'm in France, but I'm really looking forward to returning to South Africa, the place that feels like home. At the bottom of this slide, I've put on a YouTube channel link and a link to my blog site. I do that because a lot of the lectures I post that I have made available for classes have been recorded and placed on YouTube for free, and the index for them can be found at the WordPress site at the bottom. So I'm happy to make those direct links available to people if they need them, but I wanted to make sure people know that those materials are available. So I'm going to start with a basic question, really. Why should LC-MS/MS facilities, people doing proteomics or metabolomics, really, why should they need to assess quality in their experiments? And what does it mean to assess quality in their experiments? So for that, I'm going to walk through the 7 basic tools of quality as presented in a rather classic book on quality control. But for each of these basic tools of quality, I'm going to try to link them to how these tools play out when we're working in proteomics, and also try to tie these to different recommendations that people can use, especially if you are running a core facility, to ensure that the product of data that you return to facility users come with a package that accredits those data that says these are solid and worthwhile. So I've drawn a bundle around those last 3 points because they're going to be sort of tied together, but we'll bring it all together at the end, as you'll see. For many people, shotgun proteomics may be a bit of a black box, and that black box can occur on several different levels. I've met a fair number of bioinformaticians who are less than clear on what happens on the bench when someone is manipulating the sample to get it ready for an LC-MS/MS experiment. I've met a fair number of people who want to think of the mass spectrometer as a black box, that stuff goes in, magic happens, and out comes your data volume. And of course, there are plenty of people who don't understand how the data produced by a tandem mass spectrometer become identifications or become quantities. So I thought it would be worth talking a little bit about the first 2 aspects of that. I do a lot of talks about how to identify tandem mass spectra. I'm not going to repeat that material here, but I hope that at least by visiting the first 2 things, we can start thinking about where variability enters into shotgun proteomics. Okay, so this talk is only going to last about an hour. I want to leave a lot of room for questions. I was really pleased to see so many people had filed questions on the website even before the talk had begun. So I'm hopeful that we can cover some of those at the end. So let's start with a protein mixture. If you dump out the guts of a cell, how many proteins or how many proteoforms or how many different genes, gene products are represented in that mixture? I think that I can say without fear of contradiction that when you dump out the guts of a cell, you have more than 10,000 different proteins represented. And that's just counting the number of accessions. If you were able to detect every protein, no matter how sparsely it was found in that mixture, you would have a list far longer than what we produce with almost any proteomics experiment today. So if we think of a deep dive on a proteome as seeing the first 10,000 proteins, we'll say that's the detectable proteome. So I want you to think of each of these proteins as having some sort of structure, having some disulfide bonds, holding links in place and holding folds together just so. And so proteins are already kind of inconvenient for us to deal with from an analytical chemistry point of view. So we're going to want to cut them up into peptides, but we can't really do that until we can denature them, until we can make these complex structures unfold into kind of a loose chain. That means we're going to need to reduce disulfide bridges, probably hit it with something like urea to make it stretch out nicely, and then chop it with something like trypsin. And usually the trypsin reaction is around an 8-hour experiment or an overnight reaction. Once you've done that, you've gone from, let us say, a protein mixture of 10,000 proteins to something like 100,000 distinct peptides. We've made the situation worse, not better. However, peptides are a lot easier to work with in analytical chemistry than are proteins. So this more complex mixture in terms of numbers of different molecules represented is going to be easier for us to deal with at the stage of fractionation, liquid chromatography, electrospray ionization, and everything else that appears on screen here. So one of the first things that people try to do when they have a very, very complex mixture, like a cell lysate, is to use some sort of fractionation technique. Now, some people will separate at the protein level, for example, using a 1D gel in a gel CMS experiment. But more frequently, I think we see cases where people are separating the peptides on some property, whether that's isoelectric point or using something like a basic reverse phase separation. They are able to separate this mixture of 100,000 peptides into fractions that contain far fewer numbers of distinct peptides in each. So we need to combine together all of those fractions in order to account for the total content of the proteome. Each of those peptide fractions, each of the little Eppendorf tubes I've shown here, will then be subjected to liquid chromatography, usually at acidic pH, which is to separate those peptides on the basis of hydrophobicity. So a peptide that's just covered in L's and V's and I's is going to stick to the column a lot more aggressively than will something that's much more acidic or basic in nature, something that isn't so hydrophobic, in short. So the liquid chromatography is already representing the second stage of separation. We've got some sort of fractionation we've used up front, and now we're using reverse phase separation on the peptides in a particular fraction. As the peptides elute from the column, they are subjected to electrospray ionization. Often that means we're zorching them with something like 3,000 volts as they come out of electrospray, and that creates this spray that allows these peptide ions to separate from each other and to desolvate. In other words, to become sort of naked peptide ions flying into the mass spectrometer. The mass spectrometer environment is a high vacuum, generally speaking. So we want these ions to be well desolvated as they come in, and for just the naked peptide ion with, of course, the proton riding shotgun on it coming into the mass spec. In general, we are performing in shotgun proteomics 2 kinds of mass spectrometry. One is usually to do a high resolution mass spectrometry experiment where we simply ask which ionic mass to charge values are available to sample right now at this instant in time. A single LCMS-MS experiment will typically last like 90 minutes, of which at least an hour is a slow gradient as we increase the hydrophobicity. So at a given moment in time, we need to know which peptide ions are available right now. And by measuring them at high resolution, we're able to, first to resolve them, to be able to say these 2 ions have very similar mass to charge values, but we can tell them apart through the resolution of the mass spectrometer, but also to be able to say which ions are most intense compared to to all the other stuff below them, and to be able to see the separation of isotopes for them as well, which allows us to say, if we're only interested in examining +2 or +3 ions, we're able to tell the instrument, acquire tandem mass spectra only from those. So that's rather powerful. It's doing a lot of work for us. So this high resolution mass spectrum is characterizing the intact peptide ions, not proteins. They've already been digested, and not all the peptides at once, just the ones that are coming off the column right now. But the rest of this slide is devoted to tandem mass spectrometry, which is to say that we're going to isolate ions of a particular peptide. Now, we didn't tell the mass spec in advance which ones to sample. It saw that a particular peptide was available at this moment in time and was relatively intense in the mass spectrum. So the ion is that ion has now been targeted by the mass spec to collect a whole bunch of ions in the trap, to cause it to collide with noble gas atoms, which cause it to gain energy until the mobile proton causes it to break into a B ion and a Y ion, most typically. And then those fragment ions, all the fragments that resulted from blowing apart a particular peptide ion, can all be cataloged in a single tandem mass spectrum. Now, I've drawn a green line in that tandem mass spectrum over at the right, and that was not a mistake. That green line at the lower right part of the diagram is to indicate that we know the mass to charge of the ions we destroyed to create this tandem mass spectrum. So we're going to keep track of which fragments belong to which precursor ion, which intact peptide ion. Okay, I realize that's a lot of material to go through. If you've never done any work with tandem mass spectrometry, this is just an intro, and there are a lot of lectures out there that can help you get up to speed on this. You don't need to understand every last detail for the rest of the talk to be meaningful, but I wanted to give some background for people who have not done proteomics in the past. All right, now the principle at the top is true for anything in biotechnology. And here I'm going to try to unwrap that statement in the bullet points below in the context of shotgun proteomics. But at the moment, I just want you to internalize that the more complex a process is, the more doors there are for variability to enter the experiment. So if you were building a car, there could be a problem with the gear shift. There could be a problem with the lubricants you place in the system. There may be problems with the door handle, right? I mean, the more complex an object, the more ways it can break, right? The same is true for processes. When we have a complex process that gives us our data, there are lots of opportunities for variability to enter that process and give us a variable outcome as a result. So let's consider this in the case of shotgun proteomics. We already talked about the processes of extraction, where we try to get the proteins out of cells. Perhaps we've used something like enrichment. Maybe we were interested in phosphopeptides, so we used a column to enrich for phosphopeptides. We maybe used fractionation for a very complex proteome. All of those different processes can bring variability along with them. And even if you started with exactly the same blob of cells, the proteins that come out will differ if you extract this aliquot and then do another aliquot differently. Even if you try to follow the same steps as identically as possible, variability will still creep in. The way in which you do protein denaturation may have some effects. Reduction in alkylation can be quite variable. Sometimes people use just a little bit too much dithiothreatol or DTT, and this causes the peptides to get labeled in ways they didn't expect. Or maybe they handled the digestion differently. Maybe in the one case, someone started the digestion process, went home, slept, woke up, and came in the next morning. And maybe in the next case, they started it when they arrived at the lab, and they stopped it when they finished their day at the lab. These are very different time intervals. The way that chromatography takes place can certainly differ. Ionization, yes, sometimes you can have the source varying a little bit in the voltage on it, or the temperature in the heated capillary, any number of things here. The way that mass analysis occurs can certainly differ. Your instrument may be in calibration or out of calibration. The way that dissociation is taking place can change based on the pressure of gas, for example, in the place where you're doing CID. And a lot of people don't think about this, but the bioinformatics we use for making sense of these data can also lead to variability. Now, this might seem like heresy. If you have the same software and you run it the same time all over again, don't you get exactly the same result? But frequently, there are some random factors in how software proceeds in order to create a random sampling for computing false discovery rate, for example, that even if you run exactly the same search results through exactly the same pipeline, a second time you may see slight differences in which in the numbers of spectra identified, for example. So it is not the case that bioinformatics is rock solid and it can always be counted upon for giving us exactly the same result. David Fenio, I just wanted to say that this is the point where an eyebrow would raise. I know that's heresy, but I think those of us who work with software come to respect just how deeply weird it can be. Okay, so why does shotgun proteomics vary in result even if my sample doesn't? If I don't change my sample, how could it be possible that the result would change? There are a lot of factors that come together, so I've tried to summarize these again. Every proteomic sample contains more peptides than the mass spectrometer can sample comprehensively in MSMS. Yes, even if you're working with a sample that's just 100 proteins, which peptides are visible to a particular experiment will differ from run to run. If you run that same sample 100 times, each of those 100 runs is going to get a different set of peptides than did any other. It's shocking when people aren't familiar with this technology to realize just how much these data can differ on a peptide level or on a spectrum level. Certainly, potential trypsin digestion sites are cut with different probabilities. So if I have a particular protein, it might be that these 5 peptides are what I see, or it might be that these 3 from that set of 5 are observed and these other 2 are seen instead. So if you have a different probability associated with each digestion point, you get a variability in what comes out. And there are other things that can break peptide bonds. Remember, we're throwing something like 3,000 volts through these guys as they exit the chromatography, and sometimes you Zorch the peptide such that a long peptide breaks at a bond, and you now have something that looks like something other than trypsin was cutting it. And certainly in biological samples, there are other proteases that are at work. So we have to take those into account too. Every peptide must compete with every other peptide as it comes out of the column for the available protons when we are using electrospray. So something like ion suppression can occur where a peptide ion is not visible in one run, but is in another because of other peptides that are outcompeting it for the protons that are available. If it doesn't make an ion, mass spectrometry can't see it. Okay, and certainly other compounds may also produce ions even though they are not peptides. So they can create a lot of problems, especially in people working with like quadrupole style instruments. And certainly a given MS scan may represent a peptide and yet not be identified if it has too low a signal, or it is perhaps co-isolated with another peptide that appears at the same retention time and at a similar mass to charge value. So all of these things can add to this very complex result that we call a shotgun proteome inventory. Now, I've given a little bit of intro. David, could you frame one question for me about what we've covered so far? I want to make sure that that people have followed stuff, and I'm explaining this from my point of view. I'd like a question from yours if I could have one.
Yes. So one thing, could you just give a few tricks how to minimize interference during LC-MS analysis? What would be
Okay,
a trick
I'd
for
be glad
that?
to take that. Okay, so interference is the problem that you may have multiple peptides occurring at similar mass to charge values within 10 parts per million, very, very close together in mass to charge, that appear at the same moment in retention time. So in something like this, it becomes very confusing that you may have, you may be attempting to quantify a peptide in this context. And it's possible that you make the mistake that peptide A is what you quantify, but peptide B had a larger signal at roughly the same time. You see the problem here. If peptide B is absent in one run and A is correctly measured, but peptide B is present in another run, and we mistake peptide B's signal for peptide A's, suddenly we have this confusion about what the true intensity of A is. And the short version of this one is that I think we certainly benefit when we are able to do a measurement for intensity at high resolution. In the old days, we might have used something like a triple quadrupole mass spectrometer to do a quantity for something, but the triple quads are, they're not extremely mass accurate, and so you might very easily mistake one peptide's intensity for that of another. So if we're able to do something like a swath style experiment or a data independent acquisition experiment on, say, a thermo instrument, something that has very, very good resolution is better able to discriminate the intensity of A from the intensity of B, even if they have very similar mass to charge values and very similar retention times. Is that a helpful reply?
Yes, that's great. Thank you.
Okay. Yeah, I don't want to completely tell the whole story from the shotgun proteomics perspective. It's very good to be thinking about quantity as well as we move ahead. Okay, now I would like to take you back in time. I was born, I'll just share this, in 1973, which for most of our listeners probably makes me an old man. But when I was 3 years old, this book by Kaoru Ishikawa was published by the Asian Productivity Organization. Now, you might think to yourself, why on earth would a book from 1976, I mean, those are ancient times, but a book from 1976, what bearing could that possibly have on proteomics? After all, it was only, say, in the 1980s that people were doing their first experiments with tandem mass spectrometry for showing peptide sequences. But in fact, this book was transformative for lots of industries, and a whole lot of people who today call themselves quality control experts got some of their earliest training by reading this book. So I would really like to highlight Kaoru Ishikawa's far-reaching work, A Guide to Quality Control. And today we're going to talk about how it relates to making proteomics a much better quality controlled technology. And over time, a lot of the concepts from this book came to be called the 7 basic tools of quality. So I've given you the list of 7 here. Some of them, some of the later combinations of points into a basic tool are handled as separate issues, or were handled as one issue that later got separated to 2 basic tools. We'll try to just walk through this list. So we're going to discuss check sheets, cause and effect diagrams, control charts, histograms, Pareto charts, scatter diagrams, and stratification and sampling. Stratification and sampling is an obvious example of 2 very different things that have been sort of glommed together as one basic tool here, but we'll give that a pass for now. Okay, now I've done something very vile with the title of this slide. Check sheets are not just SOPs. SOP is a concept that becomes very familiar to many people in many core lab facilities, but I'm not sure that a lot of graduate students actually get a lot of exposure to this term. So I believe I've actually hidden the bottom bar of my screen so I can't see it, but an SOP is a standard operating procedure. An SOP is a standard operating procedure. Most core facilities have a recipe book of sorts that specifies when you do a digestion, thou shalt retrieve the tube from the freezer, open the tube, use exactly this much, and so on. There's a very explicit set of instructions that you follow, and you should be able to check off each one of those to say, yes, this step has been successfully carried out on the sample, I'm moving on to the next step. That's a standard operating procedure. How would you feel if you sent 15 samples to the core facility and they represented 3 cohorts of 5 samples each, and the lab used exactly the same set of procedures for the first 10 samples, and then changed to a very different set of rules, of protocols for the last 5 samples? You'd be pretty mad, wouldn't you? Because if you wanted to make a comparison to say that this protein differs from samples A to B to C, and they'd use a completely different protocol to analyze the 3rd set of experiments, you would no longer be able to make that comparison because they introduced this new factor, a new protocol. So yes, standard operating procedures are essential to producing good core laboratory science, but check sheets are not just that. They cover a lot more. And in fact, Ishikawa goes through quite a lot of detail on what kinds of check sheets he has in mind. So production, process, distribution checks. Seems like kind of a long name, doesn't it? But in these, we have some sort of value that our experiment has produced, and we want to determine, is that value in bounds or out of bounds? Is this acceptable or is this not acceptable? So is the distribution of this metric within specification? So maybe we've computed the TIC level for each of the raw files that were produced for an experiment, for all 15 of our samples, for example. We then might ask, are these TIC values, the total ion current, is that total ion current acceptable? Is it too low, meaning we might have either injected too much in the instrument, or the instrument might not be seeing it as well? Or is it too high? Have we loaded so much into this that our chromatography is going to be terrible? Measuring TIC is valuable for that. What about the charge state distribution? In shotgun proteomics, we most frequently identify +2 and +3 peptide ions. Something that's a +7 peptide ion is generally not identified very successfully. And if you had a whole bunch of +1s showing up in your experiment, you might be pretty worried that you had a bunch of ions being collected for tandem mass spec that shouldn't be, that aren't even peptides. So the Z distribution, the charge state distribution of your precursors, is useful to know for that. Maybe you're running a standard on your instrument. This is not a sample for a user of your facility, but rather it's some sort of quality control sample. Maybe it's a digest of bovine serum albumin or something like that. You might ask, what are the retention times of the peptides you see? If those retention times fall within 2 minutes of when they're supposed to be, you might say, my chromatography is great. Okay, so that's a production, process, distribution check. We can also think about this in terms of the failures of our experiments. In this case, we're doing a defective item check or a defective item location check. So in this, we might ask, how many of each defect did we observe, and where did we observe these defects? So one of the first things that we think about would be the problem of missingness. A lot of times people will do a quantitative experiment sort of through shotgun experiments. They're not targeting a particular set of ions they want to measure. They're just trying to quantify everything they identified from their experiments. Max Quant is very, very popular for this purpose. So you might try to integrate how much intensity did I see for every peptide in every experiment. That's a big challenge, right? But the software can take it on. One of the chief problems that we run into, though, is that sometimes the software tries to integrate a peptide in an experiment where it was not observed. As a result, we have a missing value. We have an intensity for this peptide in all the experiments where it was identified, but in this experiment where it was not, we have no intensity. So that gives us a missing value, and the more missingness you see for a peptide, the harder it is to do any sort of meaningful statistics with it. So we might consider those missing values as defects of a sort. Sometimes, however, our defects are much more visible. Something like a column clog could result in an experiment being improperly truncated. I said these things are supposed to last like an hour of shallow gradient on an LCM-SMS experiment. It might be that the last 15 minutes are gone simply because the column blocked up and you got no more signal for the rest of the experiment. You would want to know if that happens. And particularly, you would like to know, where did this problem take place so you can prevent it from affecting further experiments. Defective cause checks are also valuable. I'm going to show you an image of that on the next slide, so we'll get to that in just a moment. But in this case, we would like to seek the relationship of defects in our experiments to a particular instrument or a particular operator or a particular time. I'm just going to use this as a matrix, as an example. I'm not saying that you should always try to figure out who to point the finger at in the lab to say who broke the instrument, but we'll incorporate operator in this one. And finally, we have check-up confirmation checks, which is a comprehensive inspection of the final product In this case, the data set and the identifications and the spreadsheets and so on that you're going to send to the core facility users, a comprehensive inspection. So let's look at a defective cause check sheet. In this case, we have been tracking a couple operators on a couple instruments 5 days a week for the last several months, okay? I always find South African names get kind of left out of typical slides. So instead of Alice and Betty and Charla, I'm going to use Bongani and Tendeka because I knew a great guy named Bongani and I knew a great gal named Tendeka, and I really think their names should be used. So we're including them. So we have two different instruments. Both people used each of those two instruments over the space of these 6 months. And then I asked, on which days did each of those operators on each of those instruments have a failed run at some point in the last 6 months? Well, Bongani and Tendeka are really good. So generally speaking, they don't produce errors. But in this case, we can ask, did Bongani or Tendeka produce more errors on the instrument? So we could simply count. There are 3 errors for Bongani underneath Q exactive, and there are 4 errors for Bongani under the Fusion Lumos. So in total, he had 7. Tendeka, on the other hand, 1, 2, 3, 4 errors total. Okay, so we could say tentatively that Bongani had slightly more errors than did Tendeka. Did the Q exactive produce more errors than the Fusion Lumos? Well, we had 1, 2, 3, 4, 5, 6 on the Q exactive, and we had 1, 2, 3, 4, 5 on the Fusion Lumos. So the Q exactive was very slightly more troublesome, but not much more. But let's think about this from the perspective of day of the week. On Monday, we had 1, 2, 3, 4, 5, 6 errors for Monday, 1 error on Tuesday, 1 error on Wednesday, 1 error on Thursday, and 2 errors on Friday. So I think what may be going on for our errors is that something is happening on Mondays. Now, it could be that the operators come in on Monday and the operators are more prone to making mistakes on Monday. I know I am more prone to making mistakes on Monday. Or it could be that something is happening on the weekend with our instrument. What if the instrument lying fallow for the weekend causes it to be more likely to fail for us on Monday? Either of those two possibilities could be an interpretation. We would like to know which. Okay, so that's an example of a defective cause check sheet. I hope that's a nice, simple example. You can use it in lots and lots of different contexts, of course. What about a checkup confirmation check sheet? This is the final check of the product we're going to send back to our users. Were all SOP steps completed? Did we record all aberrations against those SOPs? All places where people didn't follow the SOP completely? That's an important thing to track for these experiments. And it is important to realize that sometimes we don't follow the rules exactly for real reasons, and tracking those is also important. Okay, so somebody sent us so many samples and they asked for us to use 10 fractions on each one. So I should be able to multiply the number of samples by the number of fractions to get the total number of LCMS-MS raw files that were produced. It may be, however, that I forgot one of those files. So it's a good thing to go back and check that the number of files in the manifest equals the number of files that you are transferring to them. Have we tested for potential outliers? This is an interesting problem and one we're going to look at in a very brief demo I'm going to do after the talk. Quality control sample data accompanies experiments. This is kind of a controversial thing. A lot of time, a proteomics facility will perform checks of the instrument with a digest of bovine serum albumin or with HeLa or yeast or something like that. And frequently, they don't include those data when they pass you the data for your experiment. You as a user might be interested in those though. So we should at least let the user know that these files exist if they're interested in seeing them. Because you'll be surprised how often it is that a user comes to the core facility and says, my samples were great, but your data look terrible. It happens all the time. Anyone who's worked with a core facility probably just winced and their heart shrunk a few notches as I said that, because it happens all the time. So yes, having quality control data to show that the instrument was fine, the sample, however, was not working well with our technology is definitely worth being able to demonstrate. Okay, I'm giving an example of a quality control metric for shotgun identifications here. We computed the fraction of MS-MS scans that we identified. So maybe we collected 10,000 tandem mass spectra in each of our experiments, but maybe we identified only about 2,000 of those 10,000 tandem mass spectra. That's an identification rate of 20%. In some cases, like biofluids, if someone was shooting plasma or serum, identifying 20% would be a miracle. It would be a miracle, and everyone would be writing to you trying to figure out how you did it. On the other hand, if you're shooting something like bacterial guts on your instrument and you had a huge amount of sample to work with, identifying 20% would probably be kind of disappointing. So being able to compute the fraction of all spectra that were satisfactorily identified is rather important. And finally, a service that frequently falls upon core facilities, but is not one that many of them relish, is that the data need to be made publicly available when the manuscript is accepted for publication. For that, you should be able to report a proteome exchange accession record. So if the core facility is doing that, it's important that that information be made available with the manifest. You might think, oh my goodness, I don't want any core facility posting my data. It's going to take me a long time to write the paper. But I would note that Proteome Exchange certainly gives you the option of posting files privately that are passworded that only become public when you say so. So that way you can already have your Proteome Exchange number ready to go when your paper is accepted. Okay, let's move on to the next slide. So a lot of times, the name Kaoru Ishikawa will be associated with fishbone diagrams. They're often called Ishikawa diagrams. These are intended to show causes and effects. And one of the best things that you can do when you're in a busy group of people trying to discuss what's going wrong to cause variability in our experiments is to record that information of what everyone's saying in a fishbone diagram. So first, we ask, what are the main factors that contribute to a quality characteristic? Now, Ishikawa recommends that these include raw materials, equipment, the method of work, like the protocols, and the measuring method, like the instrument. In this case, the diagram I'm showing over at the right was from a paper by Wout Bittermue, working with the Quality Control Working Group. We separated it into sample preparation, liquid chromatography, mass spectrometry, and bioinformatics. So those are the main factors that we've used in this case. And then once you have a main factor, you can subdivide that into others. Sample preparation is not a monolithic process that we go through. It's something that includes the key step digestion and the key step modifications. It might be that we are using some sort of enrichment, as I mentioned, to look at phosphopeptides. That would be an example of a special form of sample preparation. So in each case, we're going to try to subdivide these main factors into detailed factors. And we might go even further. We might separate the detailed factors into even more detailed factors. So all of these are feeding into one effect. The main arrow there points all the way to the right to variability. So you should have a very clear question that animates the creation of this fishbone diagram. In this case, what is causing the results of our shotgun proteomes to vary in the proteins that they observe and the, let us say, rank ordering based on intensity, something like that. Okay, so being able to understand that complex causes lead to a combined effect is an important factor that we just have to take into account. The more complex the process, the more complex the more doors are open for variability to enter. Okay, now the process control chart is something that was really embraced by Dr. Schuart. I've forgotten his first name, I believe, but the process control chart is a very standard way to evaluate the variability of instrumentation, for example. So a control chart illustrates whether each of the points on the graph is normal or abnormal, thus revealing changes in the process from which the data has been collected. So let us imagine, first of all, that we're trying to summarize a day, and maybe each day is going to be another point along the x-axis here. So maybe I've performed 5 QC experiments today on the instrument alongside all the experimental stuff we did. So I can say, what was the, just to use an example, what was the median number of peptides, of distinct peptide sequences that I saw from our QC standard today? I have five raw files. Each of them has a different number of distinct peptides observed. I'm going to take the middle one, or I'm going to take the average. I guess in the Schuhart case here, they use the average rather than the median. I'm going to post that average as that day's value on the top. Now at the bottom, I'm going to characterize something else. It might be that those that the 5 measurements I made of how many distinct peptides are in our QC sample gave very, very similar results, in which case the average is a very good representative of all 5. They group together very closely. They have a very narrow range. That would mean that they have a range close to 0. On the other hand, I might have had 5 QC experiments that have a very sizable difference between the highest value observed and the lowest value observed, in which case the range is really big. So in a case like that, it might, from the average, I might think I was doing fine, but the variability I observed, that the range that is incurred, is very great. So in the original process control chart shown in Ishikawa's book, we not only plot the average in the top line, but also the range, the highest value minus the lowest, in the bottom. Now you'll note that there are some values there for the average of averages at the top. That's the X with 2 bars over it, and the average of ranges. That's the R with a capital, the R with a bar over it. Okay, so that's the typical value for the averages and the typical value for the ranges. But we also have these values of upper and lower control limits. That's a UCL and an LCL. So in this case, we have been able to look at the performance of this process day after day for a very long time. And so we have a very clear notion empirically of what a high value looks like and what a low value looks like. In this case, upper control lines and lower control lines act as our bounds of okayness. If the average falls above the upper control limit, we say it is out of control. If it is below the lower control limit, we say it's out of control on the low side. You'll note that range has no lower control line. And the reason for this is that the ideal value for a range is 0. So we don't punish things for having a 0 range. Okay, so the Schuhart process control chart is basically a building upon this one that was made available in the Ishikawa paper book. All right. So what am I going to use a process control chart for? One of the best uses for it, David, I saw you moving, so if you have a question, by all means, okay, great. So for examples of process control charts, one of the things that we're most typically going to do is longitudinal monitoring, which is to say, we want to create an historic record for how this machine performs across a long spate of time. How long does your institution own a mass spec? Do they use it for 2 years and then throw it away? No, because instruments are expensive. We don't do that. But if you have $1 million instrument, you should hope, I think, that it is useful for 10 years. Okay, let us just say 10 years, but typically I think even orbit traps are not something that we expect to last much longer than that. Let us say then that for a long period of time, for many months of time, we want to track how this instrument is doing in repeated experiments, usually on a quality control sample, one that has unchanging content. Okay, so if we have particular peptides that we're monitoring in there, we might want to measure the retention times of these peptides that very reliably show up. Those retention times tell us that liquid chromatography is producing very similar gradients of hydrophobicity over time, and that the peptides are eluting right when we expect they should. That's very valuable if you're trying to do something like match between runs in MaxQuant. If you tried to switch columns in the middle of a MaxQuant style experiment, you would probably have big problems because the retention times would no longer match. It's an important factor to keep in mind. What if we were trying to do quantitative proteomics? In a case like that, we might focus on the label-free quantitation intensity that we had from a particular protein spiked into experimental samples. So maybe our samples are trying to teach us something about Mycobacterium tuberculosis guts, but we've spiked E. coli protein in there in order to make sure that we know exactly where the retention times are placed, and to be able to characterize the quantity is not changing as a characteristic between these samples. It might be that you're simply trying to ask how many distinct peptides have been observed from a complex quality control sample, a quality assessment sample. It might be that you're measuring the environment of the instrument. You might measure the voltage of the instrument or the temperature inside the instrument. Those values can certainly have bearing on what kind of measurements come from it. And certainly you'd be well advised to track the mass accuracy that this instrument can provide over time. So these are things that could give you process control charts that are very valuable to understanding whether the instrument was behaving itself or not. I've mentioned at the bottom, and again, it's hidden by the Zoom bar for me, but Hotelling created a strategy by which you could combine together different kinds of metrics and track those as over the course of time. So maybe you have 20 different features that you're measuring for this instrument, and you're able to put them all together, thinking that if something goes wrong in the instrument, multiple measures might together vary to reflect that something has gone wrong. I think most everybody knows about histograms at this point. I just wanted to point out, though, that in 1976, when the histogram was first put forward in the context of quality control, Ishikawa did not have access to spreadsheets. Things like VisiCalc and so on were just not that widespread in 1976. So histograms were still something that you computed by hand. You had to figure out where the bin sizes were. You had to figure out how many values fell in each bin and so on. It was a lot harder in the old days to do stuff like that. So when we use histograms today, we're able to do so with quite a lot less labor than what was necessary in 1976. Just the same, Ishikawa had a lot of really good thoughts about how you should interpret whether a histogram is giving you good news or bad. So what is the shape of the distribution? I think a lot of people in science frequently make the mistake of believing that all distributions in their heart of hearts are normal distributions. And this is not the case. There are many times when we'll find that, well, just to make it topical, to say, for example, that a TOF might tend in the upward direction of mass error rather than down. So in this case, its mass errors would more likely rise than fall, and so you wouldn't see a symmetric Gaussian distribution, but rather something that pushes over to the side. So the symmetry can help you see that. The skew, which is to say, is it centered or is it pushing left or pushing right? Or the bimodality. Bimodality is a very big deal. I'll give you a very simple example of a case where we think about bimodality. What if we were to write down the height in centimeters of everyone attending the call today? We have 144 participants. We just lost one, sorry. We could write down the height of all 144 people, and we could then make a histogram to say, what are their heights? And once we took age into account and stuff like that, we would probably see that we have two distributions of heights, that we have an average height here and an average height here. That's because we have both men and women attending today's seminar, and generally speaking, we find that their heights are different. That creates two different common values, modes, two different modes in the data, which is a bimodal distribution. So you have men's heights and women's heights mixed together. That causes two humps in the distribution of our heights. Okay, if that happens on something like mass error, you might ask why. It might be that you're mixing together different experiments that have different kinds of mass error in them. Okay, what is the relationship with the specification? Remember that one of our checklists, our check sheets, was to ask, is this value within the specification or does it fall outside the specification? Does this fall right at the expected value? If we have the distribution, is it, do we find that the center is where it should be for this particular metric? Maybe a mass accuracy difference of 0 for the observed versus the theoretical mass of peptides, for example, as we see over at the right. Is there a need to change the histogram? Have you got, is it necessary that you separate out multiple distributions, which is what we call stratification by factor? Or do we have so few values in our bins that it's not a very informative histogram? There are plenty of ways that this works out. So in this case, I've used a diagram from a Waters white paper that showed the mass accuracy of their instrument for a bunch of peptides that they'd identified. It's a very common example of histograms that we see in shotgun proteomics. Now I have to say Pareto charts are one of my favorite diagrams. And I think most of it comes from what we often call the 80-20 rule, which is to say that 80% of the time, 20% of your causes lead to the mistake. That is to say that some types of faults are far more common than others. There's another rule about this. When you're writing papers as a graduate student, writing the first 80% takes only 20% of the time. Sorry, this is a very unfortunate thing to hear sometimes, but it's often finishing the paper, doing the last 20% of it, that takes all the time. So the Pareto chart is kind of a take on that. In this case, we simply want to ask what contributes the most variability. In this case, I'm showing an actual Pareto chart, this one produced by Mike Berriman. Sorry, I always get sad when I present this slide. Mike Berriman was a great researcher in our field and died rather young, very sadly, in the last couple of years. But he was one of the people in the American Society of Mass Spec who really pushed for people to start thinking about quality control in a quantitative way across ASMS. So I think it's only appropriate to keep this slide in here. So thanks to him for this. In this case, he was evaluating a sample that had been analyzed many, many times over in SRM types of experiments, in selected reaction monitoring or targeted quantitation experiments. So in this case, he had asked over a large number of raw files, how frequently did we decide something was at fault based on the retention time seeming wrong for a peptide? And you see that there were 30 non-conforming cases that fell into that case. How many times did we reject a quantitation because the peak area didn't look correct to us? Maybe it was unable to get a decent peak area because there was no signal. So that accounted for the next largest number of peak fails. Then we have something called FWHM. That's a little cryptic, but it's a term that shows up very frequently when we talk about quantitation of peaks in mass spectrometry. It stands for full width at half maximum. Full width at half maximum. So if you have a peak and you want to say how wide is it, you could ask, where does it first peak up above the noise and when does it disappear into the noise again? But those bounds are really hard to find. You might say, what is the area of the whole thing? You could do that, but the peak area is a different factor. If you're trying to ask, how wide is it? The most common way that people do this is they ask, how tall does it get at the maximum? Then they pick a value that's half that height and ask, where does the peak cross upward through that line and downward through that line? That width is called the full width at half maximum. So sometimes they were rejecting peaks on that basis. Sometimes they rejected peaks on the basis of symmetry, to say that they didn't have a shape that corresponded with what they should. So the Pareto chart is always going to sort in such a way that the most common reason for failure is at the start and the least common reason for failure is at the end. We also have this little line that's rising from the top of that first bar. In this case, we're trying to ask what fraction of all errors have we accounted for when we include just retention time, and of course it's the same as the height of the bar. When we add retention time to peak areas, and we see that that rises a little bit. When we add retention time peak areas full width at half max, we have even a bit more rise to it. And finally, what happens when we take all 4 of these cases into account? Now we've accounted for 100% of our error. Okay, so the Pareto chart, the bars are showing us basically a popularity contest, which factors failed most frequently or caused the fault most frequently, whereas the line is showing us what fraction of all fails have we accounted for so far. That is a Pareto chart. Scatter diagrams are certainly very, very useful. Certainly with spreadsheets and languages like R, they've gotten a whole lot simpler to work with as well. And so for this, I wanted to give an example of an XY scatter plot in the context of quality control. In this case, Vanderbilt University was taking part in the CPTAC network, analyzing a whole bunch of colon tumor samples. We had, let me remember this correctly, 15 fractions for each of 95 samples. It was a lot of data, took us 8 months of time on the instrument. And so we wanted to track over time whether these data looked like each other, or did we see a strong change in how the instrument presented data from month 1 to month 8. And there was quite a lot of walk around within this space, such that we see that when all of the raw files for the black set, which I believe was the first one we ran, and the deep red set, the second one we ran, tend to separate from everything else. This is an example of instrumental drift over time. And it's worth keeping in mind that if you're analyzing samples on your instrument, you should not expect that shooting a sample this month and next month is going to give you identical results. It's not a reasonable thing to expect of this sort of sampling technology and this very complex technology. I see that a QA question has appeared. Okay, I'm going to come to that at the end if that's okay. So we'll circle back to the Q&A session after this. Okay, thank you for filing that though. Okay, so why do we need to use XY scatter plots? Ishikawa had several things to discuss about this. His first was that he wanted to relate a cause to an effect. So if you had an X coordinate that reflected, say, the temperature of a part of the instrument, and a Y coordinate that said, how many of the spectra did we identify? You might be able to make the argument that that if there's a strong relationship between these two values, that one is causing the other. That's actually something that most statisticians would frown upon quite significantly. Just seeing that two values rise together and fall together is not enough to say that one causes the other. Okay. It is, however, quite useful to discover covariance, correlation, or association. Each of these 3 terms means something slightly different. We're not going to get into it right now. But probably the most common use that we see for scatter diagrams in the quality control domain is that we want to visualize something from principal components analysis. Principal components analysis is very useful when we're trying to boil together a lot of different information from these experiments into a single axis. So in the case of the Vanderbilt instrument diagram at the right, the first principal component is the one that explains the most variance across all of these experiments. The second principal component is the one orthogonal to that accounts for the second most variation overall. That's what principal components analysis gives us. Oh, it's because I clicked on the Zoom interface. I got to get back to the slideshow. Here we are. There we go. Okay, stratification and sampling is a rather complex topic, but I think it's one that we can cover at least at a very high level. If your samples fall into different categories, stratification is a way to incorporate those factors of which category they fall into as part of the model. So I already made the example that if we were to write down the heights of everyone on this call, we would need to take into account the age of the person and the sex of the person and so on in order to understand why some people are not as tall as others. So on the right, I've got a real world example from a paper I wrote for the CP Attack Network many years ago. In this, I was asking about the repeatability of identification of peptides. Remember, I said that even if you shoot the same sample many times, which peptides you see from it is going to change each time. So in this case, we have instruments at several sites, site 56, site 65, site 86, and site 95, and site 73. They're a combination of Orbitrap instruments and LTQ instruments. And in each case, we're asking what fraction of all of the peptides identified were observed in all 3 replicates, in 2 of the 3 replicates, or in 1 of the 3 replicates. So you see that the blue bar is the number that we're seeing in all 3 of the replicates. The green is 2 of the 3, and the yellows are the 1 of the 3. At the left, we're looking at fully tryptic peptides, which is to say that this peptide conforms with what trypsin should be cutting on the N-terminus and what it should be cutting on the C-terminus. A semi-tryptic peptide, on the other hand, is a peptide that only conforms to trypsin specificity on one end or the other, not both. What we see is that tryptic peptides are far more repeatably measured in a shotgun proteomics experiment than are semi-tryptic peptides. So if you are talking about repeatability in the abstract and you don't separate these classes of peptides from each other, you're actually mixing together 2 very different trends. So we need to stratify them in order to show their independent effects. Sampling is, again, a fairly complex subject, but I really love this quote from Ishikawa's book. Since populations display this frequency distribution, you must be careful to maintain a strictly random sampling. In other words, do not just select or pick up the good or bad pieces. Don't just take samples from only one portion of the lot. The samples must be truly representative of the lot. If you are not, if you try to learn how to keep your instrument running well by only looking at bad data, at failed experiments, you're missing half the story. A well-run experiment is not just one that is free from whatever caused this experiment to fail. We need to be able to think about all of the data, not just those of a particular set, if we're going to understand its proper use. Okay, this brings me to my last slide. Several takeaways that I hope everyone can grasp right away. The 7 basic tools are intended as a guide to action. This is a bit of a challenge, actually. Imagine that you ran quality control samples on your instrument every day, but you didn't bother to come back to those data sets to understand the typical performance of your instrument. What if you simply open a raw file in the instrument control software and say, yeah, that looks pretty good? Have you quantitatively characterized why it was good? Have you said what was the intensity level? What were the retention times like? What fraction were identified? We want to be able to guide action. So when we see a check sheet value that falls outside the realm that it's supposed to be, when we see that one cause is leading to almost all of the failures of our instrument, if we don't use that to guide our actions, we're losing the value that experiment could actually pose for us. So when you do when you make use of these 7 basic tools, you are in fact telling yourself what to do in response to see that your instrument continues in good health. Okay, these tools were all defined long before MS proteomics was a thing. And yet, these same tools are still quite relevant in guiding its development. In fact, I'm going to go further and say they are not routinely used by many people who really need to be using them. So if you're joining a core facility and you see that this is not something that is taking place, it's a good idea for you to go ahead and start creating standard operating procedures. to create check sheets. Very frequently, when a graduate student joins a laboratory for the first time, they are told to ask all their questions to the postdoc. And the postdoc is there for 2 months and then they're gone. And someday the grad student has a question that they cannot ask the person who's no longer there. We need to capture this quality control information, the standard operating procedures and so on in an institutional wisdom kind of way, in a way that we're able to all refer to it and all agree on how we shall conduct our experiments. All biological mass spectrometry facilities should incorporate QC in their everyday workflows. We need to ensure that tomorrow's processes are based in careful audits of yesterday's experiments. At present, a great deal of this is taking place in a very ad hoc way, and I think that as we become more structured about how we characterize past experiments, we will get better at making sense of them. Okay, David, I'm going to turn the mic over to you.
Yes, thank you very much, Dave. This was excellent. This is really, and as you say, I mean, I agree completely that not enough labs are doing this. That's unfortunately very clear.
I have a small demonstration with some software a graduate student of mine developed that I think can show a little bit of, can shed some light on the outlier problem. So we can pause for questions here, or we can move directly to that, either way.
Maybe let's take a little bit of questions and then we go back to your demonstration. Maybe let's start with this. There's 2 questions in the Q&A. So let's start with the, I have soil metabolome samples collected in batches. I'm planning to analyze immediately as they're collected because the number of injections are large. In this case, what could be the best QC approach?
That is interesting. I think that AJ Sorti, I see is the name there. That is an interesting problem. When you are conducting a large-scale experiment that spans many weeks of time for the instrument, you really need to be very careful of what's called a batch effect, which is to say that if you queue up samples for a month and then start them shooting, collect samples for another month, and then send those over to the lab for shooting, and then collect samples for the 3rd month, and then send those over to the lab for shooting, you are quite likely to see that the experiments from batch 1 are not comparable, or cannot be compared to the samples from batch 2 or from batch 3. Finding a way to control for this batch effect is probably one of the most valuable things you can do. I would urge you to pick a sample for which you have a lot of extra quantity at the start and make the decision that you are going to make sure this sample appears in every batch. When you do that, you're able to compare a sample of known content that appears in each of the batches to evaluate how much shift you're seeing in instrument performance between these different time intervals. It is possible to do an experiment of this scope. I would note, however, that you probably cannot expect retention times to be well conserved from one batch to the next, because it's possible that even the column would change in such an interval. So I would be careful of that. Be able to control for the batch effect by having a sample that appears in multiple batches, and make it a factor in the statistical model that you are using to characterize the results. Good question.
Great. Yeah, no, that's great. Yeah, so we have another question regarding effect of ionization mode on the variability of the final data. And this is looking at different HCD, HD plus C, ETD, and so on. So several different ionization modes, and how they would affect the sequence coverage and detection of low abundance protein, in this case in biological fluid like lymph plasma and cerebrospinal fluid. Do you recommend the standard protocol, or would you how would you decide between these and apply the QC methods to decide?
I would say that many people hobble their experiments by trying to answer too many questions in a single study. Every added question that you put on top of the first one can kind of cut into the power available to answering the question. So if you are looking for a difference between, sorry, this isn't the soil effect, this is a biological fluid, okay. So if you're trying to ask, how do lymph, plasma, and cerebrospinal fluid differ in the proteins they contain, that's a biological question. If you're then asking, does HCD detect different protein differences than CID does, or than ETD does? Suddenly you've got an analytical chemistry question superimposed on a biological question. HCD is a higher energy version of CID, so it doesn't really seem to me that there's a huge amount of value in comparing the two of those. One is carried out in the trap, and one is carried out in something more like a beam within the C-trap. So I think that it's best to figure out which ionization or which dissociation method you want to use from the word go. For this, I would say, if you're looking at intact proteins, if you're doing proteoform top-down mass spectrometry, something like ETD or ECD has a lot of value. If, on the other hand, you're looking at peptides, if they're likely +2s and +3s, there's really no reason to expect ETD to be of much value. ETD becomes more valuable as the charge state goes up on what you're analyzing. So it's much more useful in a proteoform than it is in a peptide. So I would be careful to tune your experiment to the question that you want to answer, the one that gets you graduated. Frequently, we have to be careful that we don't undercut our experiments by asking questions in too many dimensions of the same set of samples. Good question again.
Yeah, great. So let's see. So I've seen some people raise their hand. So how does that work? Can we have people ask questions, unmute themselves and ask questions live if they raise their hand?
Yes, definitely. If anyone has questions they want to ask in person, please go ahead and raise your hand. We saw some raised hands earlier.
But they're gone now, or?
They're gone now.
Ah,
yes.
okay. Yeah. So please raise your hand, and then we will, two. Now we have two. So Poonam, I think you can unmute yourself and ask your question.
No unmute option for audience members.
Yes, I think you raise your hand, and then you will be moved to a panel, become a panelist, and then you can unmute yourselves. I believe. So I'm going to, Poonam, can you unmute yourself?
Thank you for the nice things you're saying in chat, by the way.
Yeah, I guess, yeah, so Poonam, while you try to unmute, let's we have another question here. So I have two batches of data related to DIA mass spec, and I'm doing biomarker discovery, but the results from each batch is different. In this case, how can I control for batch effect?
Ideally, you had a sample held in common between the batches. If you didn't, do you have a QC sample that was run in both batches? I will note that some of the most embarrassing moments in mass spectrometry biomarkers have come because folks ran all of their cases in one batch and all of their controls in another batch. You must never, ever do this. Because in a case like that, the technical variation is completely confounded with biological variation.
Thanks. So John, can you?
Yeah, I'm able to unmute myself.
Okay,
I
great.
tried also to unmute Poonam, but I don't have the privilege to do that. But actually, I just want to ask a kind of maybe a softball but philosophical question. And because some of the things you mentioned to me might occur to me rather as quality assurance than quality control. And I wondered if you could give your perspectives on if these are concepts we need to divide and how you do categorize them.
I admit to have having sort of a lazy perspective on this one, in that I think of quality control as something you sort of impose after the fact to say, how did we do? What was our scorecard? Whereas quality assurance is a process you put in place to make sure you screw up as little as possible. So my personal QA regime is that I try to wake up before 9 a.m. every day. I try to get to sleep before midnight every night. I try to have breakfast. That's my quality assurance routine. But then my quality control sheet looks like, how many times did I yell at someone today? Did I yell at the telemarketer? That was not nice. That was not nice. Yeah. So in the same way, when we have a quality assurance a program in place for our laboratories, we've taken the intellectual exercise of asking, what are the things we need to be doing to produce a quality product? And then at the end of the day, being able to audit yourself to assess whether you were successful or not in that. So that's a pretty weak version of the distinction, I think. And somebody who was a native to quality control and quality assurance spaces would probably be able to speak to that a little more directly. I've probably published 10 papers in the area of quality control and quality assurance, and I still don't feel completely comfortable with drawing a firm line between those two. It's a little like
part of the reason why I asked, because I think it's worth mentioning, right?
I occasionally see people drawing a very clear line between what is bioinformatics and what is computational biology. And I'm like, you're a filthy liar. None of us know. But I have enjoyed being on the bioinformatics side of the world, and I don't complain when people call me a computational biologist.
Fair enough. Thank you.
Thank you. Good question.
Okay, we have more questions. Or do you want to do your demo now, maybe, and then we continue after that?
I've seen a few people in the chat asking about certificates. Now, I don't have any means to do that, but if I take it that was a phone. Okay. If I don't know that Bioprotocol makes certificates that they could have me like affix a digital signature to or something, but if your lab needs a piece of paper saying you watch this thing, I'll happily scribble my John Hancock on a piece of paper if that helps. But I hope that your knowledge gained from this session is your calling card, is your certificate. But I'd have to ask our organizer if that's available.
So we can take another question about missing values in quantitation studies. And what's your recommendation how to deal with them, and what I mean, percentage would you recommend the cutoff for how much missing can still be allowed?
Okay, this is the question of missingness. Sorry. When cars drive by, I have very little ability to block it out. Missingness is a real challenge because if you're trying to do something simple like a t-test, I don't know how well many implementations of t-test work if you say that 3 of these 5 values are NA, and on the other side I have 1 NA, and not applicable. So that's likely to pose a problem. Generally speaking, people who want to use simple tests for the purpose of comparing the presence of a protein will do something basic like require all of the values to be present before a test is performed. So to think about it one way, imagine that you're doing your comparison not at the peptide level, but at the protein level. In this case, you could ask, what was the LFQ value for the protein, not for individual peptides? So it might be that in one case, the protein was observed with these 7 peptides, and in another it was just 5 peptides, in another it was 2 peptides, in another it was 11 peptides. From the protein perspective, all of those are acceptable, and you end up with an LFQ value. But it is certainly possible to have it is certainly possible to try to do a peptide level differentiation and be completely swamped with missing values. My argument is that if you're going to do a quantitative test of that sort, if you want to be able to say there's 2.5 times as much of this protein in samples of cohort B as there are in A, you really want to do a quantitative experiment. Far too often right now, I see that people want to ask clearly quantitative questions of shotgun proteomes, and that's not a very good recipe for getting a stable answer out. So if you are able to use something like selected reaction monitoring or parallel reaction monitoring or SWAT or some other sort of data independent acquisition experiment, you're much more likely to get a usable quantitative answer than you are from using a bunch of shotgun experiments. They are necessarily spreading the mass spectrometer's attention far and wide. And that's not really what you want if you're trying to get a clear quantitative result. Okay, so I would say if you're going to do a quantitative difference test in a shotgun proteome experiment, it's probably best to limit your questions to ones that can be answered at the protein level, not the peptide level. It is not completely unreasonable to throw out the proteins that have missing values. Note that you could have a protein that's present at very high values in these 5 replicates and appears not at all in these 5 replicates. That protein could be thrown out under a rule like that. So best to keep in mind some of the negative effects of that. If you have a very talented statistician around, they may be discussing terms like imputation for trying to repair these spreadsheets and fill in the not applicables with values. Most of these imputation methods have some kind of difficult properties for doing proper t-testing and stuff. So I'd be cautious in that case. They work best in cases where the missing values occur completely at random. It's an MCAR criterion, the missing completely at random criterion. If you have missing values like that, you're probably in okay condition. The trouble is in proteomics, the missing values tend to be where you have very, very low intensity, which means they're not missing at random. They're missing not at random, MNAR. And imputation doesn't deal well with that at all.
So a slightly related question that we got earlier was, I mean, you use peptide quantitation is what you measure, and then do, so what kind of QC, so one classical question there is how many peptides is the minimum? But then also, do you ever look at, is it how much variation there is between samples in which peptides you observe? Is that something you would use?
It would be possible to say the only peptides we will take into consideration are those peptides that occur in at least half of our samples. You could go that route. In that way, you might end up having only one peptide for some proteins and dozens of them for others. Ultimately, the information you can get from a peptide chromatogram inference out of shotgun proteomes is, and you can integrate the chromatogram observed for that peptide at the MS level. If you have a shotgun experiment, you have a non-zero chance of misinterpreting a different peptide's intensity pattern for a peptide of interest's intensity pattern. It's really better if you're able to use something like data independent acquisition experiments, or SWAT, or SRM or PRM style experiments, because you get measurements of each peptide at both the precursor level and the fragment level, and multiple snapshots of those fragments. And that's just a whole lot better for trying to quantify peptides and distinguish their signals from each other. So
yeah,
do I think you cannot compare sorry?
Yeah, I mean, that's great. So pretty much one thing you're saying is that first you do shotgun and get some idea, and then really to if you're really interested in quantifying something, well, you should do targeted.
But I have to be real about it. People don't.
No, yeah. No.
The number of times that many graduate students at my former university asked me, well, for this paper, I'm going to do the shotgunning, and we'll save the verification step in SRM into a separate paper. And I'm like, you're never going to do the second paper, and you're just trying to avoid doing multiple testing correction.
Yeah.
It comes up a lot, a lot. So might as well just speak the truth here.
Yes.
We should be deliberate in how we design these things.
Yes. Yeah.
Designing a quantitative study is very important.
Yeah. No, that's good.
Some journals will reject now on somebody who claims to have a biomarker strictly from shotgun data. And that's, I think, a reasonable position.
Yeah. So let's see. So there's another question about FDR, and that people often use 1% for reporting identifications. Is that, do you want to comment on that? Is that,
be happy to. Okay, I would say that if what you're trying to produce is an inventory, then it is reasonable to be very careful about the FDR of that inventory. If, on the other hand, what you're trying to find are differences, then evaluating the false detection of a difference actually becomes far more relevant. We were trying to think about ways that we could, I'm sorry, before I go too far on this, I got to remember false discovery rate gets reported at a variety of different levels, and people tend to treat them all as one thing, and they are certainly not. So the false discovery rate of PSMs, of peptide spectrum matches, is completely different than the false discovery rate at the peptide level, which is completely different than the false discovery rate at the protein group level, once you've done parsimony and grouping and stuff like that. People often talk about false discovery rate as though it's one thing, and they say it's 1%, and I have no idea what they're talking about because they haven't told me what level they're describing. In our software, we tended to control the false discovery rate at the peptide spectrum match level, and then we would determine empirically what the false discovery rate for the protein level was. That's not how systems like peptide profit and protein profit work, for example. So if you're using Philosopher or MaxQuant or what have you, you'd better understand which false discovery rate it's computing and how, and what level that it applies to. 1% is just a number. There are people I've worked with who say anything beyond 1% is asking for trouble, but the default in our software for PSM false discovery rate was set to 2% by default. So the important thing is to understand how these false discovery rates connect to each other. That controlling a false discovery rate of 1% for PSMs in an experiment with hundreds of raw files is likely to get you in trouble. But I think people don't notice that there's a difference.
Well, yeah. Do you want to do a short demo now?
Oh, sure. If I can, I'll take a quick moment with that. Let me see. I'm not screen sharing at the moment. I'm going to show you some software written by a graduate student of mine. We have a link for it that I sent in the chat way early, way at the top, I think. I'm going to try to share that user interface. Okay, share. Where's assurance? Here's assurance. Okay. So graduate student Marina Poe created this as part of, I'm sorry, her married name is Marina Creek, K-R-I-E-K. But the link for it goes to her GitHub repository set up in her earlier name. So assurance is designed to make it really easy for people to find outliers and other odd behaviors in their data. One of the things that you can do is run Quameter on a set of raw files that you have to generate a set of metrics. In this case, I'm going to use a set of metrics that I've already generated offline. So I'm going to browse to a directory containing those. It was in a short course that I just taught over at IMSC. In this case, I'm going to grab the TSV file for tuberculosis metrics. That file I also made available in the chat way up high. Hopefully that's being reshared. Okay, I click open. Now the software has picked up all these metrics that I generated in Quameter offline, and now I can run the detect outliers option on it. Okay, in this case, we had 120 raw files. It should be pretty plain that the biggest variation, the biggest variance in the data set is explained by principal component 1 here. It accounts for 51% of all variance in the experiments. And we have these 2 experiments that are way out here on the end. I'm going to hover here. So I want you to remember SW2-1-10. And this other one is SW2-1-9. So those 2 raw files are clearly well out from everything else. Now, I want to understand why those are so different. One of the things that I can do within the outlier detection result is to show the loadings to see which aspects of the PCA. Okay, that is really illegible on that very small window. So sorry. So once I pull it up to full screen, I can see that some of these metrics that I fed it are quite strongly indicative of principal component 1. So that is telling me about the nature of the metrics that make these 2 raw files look so different from the others. So I want to show you a kind of a more, a less computationally intensive route for looking at these same raw files, which is to plot individual metrics. So in this case, I'm able to look at each of the metrics that Quameter outputs. You can see that, well, let's drop down to one that's easy to understand. Here we go. This is the fraction of precursors in a given raw file that were +2 charge. Okay, so we see that at the lowest, I had 40% of all my precursors were +2s. At the highest, I had about 0.74, so 74% of my precursors were +2s. So that's an example of a metric that helps me to evaluate whether any of these files are way above or way below the others. But I wanted to show you a little bit of snooping that we did on the people who ran these data for us. You see, their raw file names don't really tell us much about when they were run. So they just gave us back files that were labeled H1, blah, 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 H2, blah, 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 SA1, da, 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 SW1, that sort of thing. But in this case, we're looking at the start timestamp, which is to say that within the raw file remembers when it was actually produced by the experiment, by the instrument. So even though the core facility didn't tell us when the raw files were actually run on the instrument, we're able to look inside the raw files and find out. And what we see is a really odd pattern. Here we have our first experiment run on the 21st of October in 2012. Here we see that the last file was run after my birthday, around Thanksgiving Day in 2012, November 25th. But I want you to note this huge gap right here between this file and that file. What we see is that on November 1st, something happened. Now, we don't know what it is. They didn't record this. We didn't get a copy of their lab notebook. But for some reason, on November 1st, they stopped shooting our experiments. And it wasn't until, say, November 24th that they got back to them. When we look at the two files that caused such, that looked so far out from the cloud in the loadings, in the outlier plot, they were numbered SW2-1-9 and SW2-1-10, these two files. So what we see is that by sniffing the data, by looking for outliers, we recognize that something was going very hairy with the instrument here on November 1st. They did something, and it was only after they did that something that they got back to running our experiments. So sometimes doing an outlier sniff of your data can tell you a little bit more about what was going on in the lab that produced them. And that is awfully valuable to a lot of people. The software is called Assurance, and it's available from her GitHub. It's currently in version pre-1.0, and I swear we'll get this manuscript published if I have to write it myself. But she has a job. She's doing very interesting things. Okay, so that's the demonstration. Did we ever find out what Poonam Sharma's question was? Because we really should know.
I agree. We should know.
Yeah.
Can somebody on the technical side unmute Poonam?
I'm not allowed to.
I can ask to unmute, but that's all we can do. I don't think one can ever unmute anyone remotely. That's what's not necessarily because if it was possible.
I'm so sorry, Poonam. Your
Yeah.
question matters to us. I just wish we knew what it was.
Yeah. Okay.
Maybe type it. It's not too late.
Yeah. So yeah, so there were some other discussions in the chat with some questions, but
I tried to just add some
Yeah.
color comments and
that's great.
information.
Yeah.
Yeah, along the way.
That's perfect. Let's see. Any other questions from anyone? Let's see here. Yeah, I think we've run out of questions as far as I can tell.
Anything in the pre-webinar list you had, or have we covered that as well?
No, I mean, we covered some of that, but I guess we can continue a little bit there. That's true. Let's see. Here's a general one. What are the common mistakes one can make with biological mass spectrometry?
Rough question.
Yeah.
I would say that people fall in love with the set of tools they have. And sometimes if you come to them with a Phillips screw, they take a whack at it with a hammer, because the hammer is what they know. People have grown very accustomed to shotgunning at the peptide level for producing these proteomic inventories. We're very good at it. And it seems like most instruments these days get evaluated by how deep into the proteome they can identify. But that's not really fair, because very frequently, the questions we want to ask are quantitative, and people do not frequently use instruments like Q-exactives or Orbitraps for doing parallel reaction monitoring or DIA style experiments. This is a missed opportunity because very frequently the questions we want to ask are known even before we do the shotgun proteome to be part of a particular pathway. And we already know what proteins fall in that pathway. We could be developing quantitative experiments that closely measure members of a particular pathway, but instead we just keep shotgunning everything. That is not an optimal place to be for finding quantitative differences in proteomics. My talk is about quality control. Assuredly, we want people to use quality control tools much more regularly. And this is, it's simply not something we've accomplished that well yet. But I believe that the voices about, well, proteomics isn't reproducible are not going to go away until we're able to document everything that we do to produce the most reproducible proteomes that we can. Thank you all for your very kind comments. I really appreciate it.
Yeah. Oh, that is great. Thank you very much, David.
It's great to hear your voice again, David.
Yeah.
I look forward to seeing you again soon.
Yes, hopefully soon. Yeah.
Yeah. Okay, great. If we think we're done with questions, I think, yeah, I want to thank David again so much for doing this today. This was such an amazing, informative talk. We really appreciate you taking the time to also answer all these questions. I think this helped people a lot. And and thanks, David Fenner, also for hosting this wonderful question and hosting David. We want to encourage people to keep posting questions. If any questions come up later, or if you think your question wasn't answered, please go to the website. We have a screenshot here. There is a large Q&A section, and you can post your questions there. You can look through questions. We will invite experts, just as both David's here, John, other experts, to answer and review those questions. We would also invite you, if you feel that some of these questions are that you see they are in your area of expertise, please answer them. We are, our goal really is to grow a community here for people to keep discussing this particular topic today on quality control and mass spec. Yes, we had several questions about the recording. There will be a the video, the talk was recorded today, and the recording will be posted later. We'll send an email once it's ready, so you will all get an email. And yes, if you want to see what's next, please go to our website, bioprotocol.org/webinars. We will have more webinars in the future. We would really appreciate your feedback. So thank you all for sticking around and joining us today, and we see you next time. Bye-bye.